Right now, millions of your cells are dividing, and each one must perfectly copy all 3 billion letters of your genetic code. We usually visualize this replication as a smooth, continuous unzipping motion, where both sides of the DNA ladder are copied in perfect unison. In reality, while one side copies smoothly, the other is built backward in jagged, disconnected chunks. This fragmented process creates a persistent misconception that the lagging strand is structurally inferior, or perhaps missing the vital chemical bonds that hold the molecule together. To see why half of your genome looks like a broken construction site, we have to look at the rigid physical constraints of the double helix. The DNA double helix is an anti-parallel structure. The backbone of this ladder alternates between sugar molecules and phosphate groups. Because of the asymmetrical shape of the sugar, each strand has a distinct directionality, designated as 5' to 3'. They act as two highway lanes moving in opposite directions. If the left flows downward, the right flows upward. The enzyme worker responsible for reading the old code and building the new matching strand is DNA polymerase. DNA polymerase has a strict chemical limitation. It can only attach new building blocks to a free 3' end. The enzyme effectively has no reverse gear. It can only travel in one direction. This creates a difficult engineering problem. The cell is forcing a machine that only moves forward to copy a track that runs in two directions at once. First, the helicase enzyme acts as a biological wedge, unzipping the double helix to create the replication fork. On the leading strand, the DNA polymerase faces the correct direction and simply follows the unzipping wedge forward in a single continuous line. On the opposite side, the polymerase is pointing backward, facing away from the unzipping fork, and toward DNA that has already been copied. The enzyme works around this by using a stutter step. It waits for the fork to open, clamps on, builds a short segment backward, and then detaches once it runs out of room. These disconnected, backward-built segments are called Okazaki fragments. This fragmented construction represents an inevitable logistical compromise, the only physical way a one-way machine can copy a backward-facing track. At this stage, the lagging strand contains all the correct genetic information, but its physical structure remains interrupted by the gaps between those Okazaki fragments. These gaps do not leave the strand structurally weak. A final cleanup crew arrives to finish the assembly, an enzyme known as DNA ligase. DNA ligase acts as a molecular glue, sweeping along the lagging strand to physically forge the final phosphodiester bonds, the chemical links that hold the DNA backbone together. This reaction permanently stitches the fragments into a single, highly stable line. Despite their vastly different construction methods, the finished leading and lagging strands are chemically identical. This elegant engineering outsmarts the strict limitations of molecular chemistry, ensuring the human genetic code is preserved